I'd like to welcome everybody to the May 1st virtual seminar of the IMAG MSM Working Group on Multiscale Modeling in Viral Pandemics and GLIMPRINT. I hope the weather is nice for everyone enjoying May 1st. I know for our European colleagues it's definitely a holiday, so if you're here today, I appreciate it. Today we have a talk from Markus Covert from Stanford, and we're very grateful to him for being willing to speak to us today. As always, I need to remind people that these talks are live streamed and recorded and made available in an edited form afterwards for archival viewing. As always, Tom Helikar, myself, Jim Sluka, and Lorenzo are here to try to take your comments, suggestions, ideas, complaints. Please feel free to reach out to any of us and tell us what we can do better. We have a variety of communication channels. They work better when we use them. Please help us let people know about these talks and their series, and also our great archive of scientific talks from past speakers. Our archive now has more than 150 talks on various aspects of multiscale modeling, often built around concepts of immune response or infection, but also general problems in multiscale modeling more generally. If you have a publication to share that's relevant to this field, you could send it to us. We could put it up on the IMAG wiki page, and we also can put it up on the GLIMPRINT page. If you have seminar announcements, meeting announcements, workshops, news items, job openings, funding announcements, we will do our best to get the word out to the community. If you'd like to sign up for GLIMPRINT, you can do that on the website. We do appreciate members helping us build the community in this area. I want to remind people that starting our next meeting of May 15th, we are moving the seminar series earlier to 10 a.m. Eastern Time. It actually says 10 a.m. EST. It should say 10 a.m. EDT. We realize that that's not so convenient for our West Coast colleagues, and we apologize for that. But we've had repeated requests from our European colleagues to have an earlier start time. And so we're trying to come up with a time that is a compromise between California time and European time. I want to make a few announcements. I remind people we have the CompuCell 3D virtual tissue modeling workshop and hackathon this summer. That's online. If people are interested, you can get information from the website and register for that. There'll also be a one-day workshop before the main Society for Mathematical Biology meeting in Edmonton on multiscale modeling, OpenVT multicellular modeling workshop. There's going to be a half day of teaching various platforms, including Iran's Vivarium and CompuCell, Physicell, Morpheus, Chaste, and others. And then a half-day meeting on reproducibility and model interoperability. And so we'd love to have people who are going to the SMB meeting participate in that. If you want more details and sign up, we can send that to you as well. Grace has asked us to let people know that there's going to be a special session on cyber infrastructure and emerging technologies at ISMB ECCB 2025. If people are interested in that, reach out to Fengluo Mao at NIH. That sounds like a good opportunity. There also is coming up in May the spring open house for Bridge to AI, also coming from Grace, asking us to let people know about this. I don't know if that exists online or if that's only in person, but if you're in the Washington area, it could be very interesting to this community. There's a new FDA initiative on new approach methodologies that may be of interest to people. This follows up on the complementary series. There also are new initiatives, of course, at NIH that have just come out in the past few days, led by Nicole Kleinstreuer. And we don't have the announcements up yet to put here, but people should keep an eye out with NIH for that opportunity as well. We are still scheduling our initial early May 15th seminar. We have talks scheduled for May 29th, June 12th, June 26th, and July 10th. July 10th, we have Jacob Barak, who was one of the founding members of this group, so 5 years ago now, who will come back and bring us back to the COVID questions that got the group started. So I hope we'll be seeing people. As always, we are looking for your suggestions for speakers. Feel free to suggest yourself, your graduate students, your postdocs. If you've spoken before and you have new information that you want to talk about, that's fine. If you want to talk about a different project from the one you've talked about previously, that's fine. If there are experimentalists or clinicians that you'd like to hear from, a paper that you've seen that's be interesting to see presented, let us know. Lorenzo is very effective at getting people to come to speak, and so please do reach out to us. This series only exists to serve the interests of the community, and that works only if we hear from you about the people you'd like to hear from. And so please do help us by telling us the people that you would really like to hear from the most. And we'll do our best to get them on the roster for you. We will have a steering committee meeting tomorrow morning at 11 a.m., and that will be to discuss integrating this seminar series with the seminar series on multi-scale modeling led from Toronto by Maria Abuchakra under the Virtual Human Development Project. Their seminars are also Thursday mornings, and there's a lot of overlap in terms of the interests, and we're thinking about trying to coordinate that so that we don't compete with each other, and so that will be under discussion tomorrow. There are a number of other topics of discussion, including the SMB organization. So please let us know if you would like to attend that steering committee meeting and you haven't received an invitation. We'll be happy to send it out to you. Again, reminding you people that starting in 2 weeks, we'll be moving earlier in the day. Are there any other announcements before we get started with the seminar? I don't want to take any more time from Markus. We're really, really pleased that Markus was willing to come talk to us today. Markus Covert from Stanford University. I think people are here because you know the work he's doing and how exciting it is. A lot of new things happening, I think, between Stanford and Connecticut and other places, which I hope we'll hear about a bit. I don't want to take any more time from you, and I will turn the screen share over to you.
So what you're looking at the left, so I'm a big fan of Andy Goldsworthy. I don't know if any of you have heard of him. He's a great artist from the UK, and he'll go out into nature, and for example, he'll find fallen leaves of all these different colors and make these incredible collages, or he'll put icicles together. If you haven't seen him, you've got to see it. It's really great. But in the spirit of his art, I asked an artist friend of mine, Eric, to come to the lab one day and make a sculpture that would kind of indicate. So what you're looking at here is a photo. There's nothing really retouched about it, but what he's done is he's taken filter tip pipettes and cryo vials and conical vials to try and build but also I should say also computational things like Ethernet cables, etc., integrating experimental and computational aspects to create a model of E. coli, right? Something that will give us a new way of thinking about things, a way to inspire us. And so I love that idea. And of course, we've been trying to build something maybe not from an artist's point of view, but from a bioengineer's point of view, and following in the lines that originally suggested by Francis Crick and Sidney Brenner walking on the beach one day in La Jolla. Um, and just asking if it was possible to have a complete solution of E. coli, right? So they wrote this really interesting perspective with a bunch of different recommendations. As far as I can tell, nobody did anything about this, but it is inspiring and it's exciting to see. And we've been kind of trying to use that as our guide, our vision for what we want to do next. And so when I'm talking about a model, I guess when I'm talking about a model, what I'm talking about is taking everything that's known about E. coli, data generated over decades in dozens of labs, thousands of papers, millions of data points generated over this vast amount of time, heterogeneous in nature, right? Large and small, somehow bringing them all together using the language of mathematics. And so you can imagine, for example, building a model of all the RNAs in the cell, or using equations of all the RNAs in the cell to incorporate and encapsulate a bunch of those data. For the 1,500 small molecules in the cell, the same thing. You can write a set of equations that generates how they come and go, and the same thing with the 4,500 roughly proteins in the model or in the cell, trying to account for all of them. So if we take all of that and then group these things into their functional categories, this is where vivarium turns out to be very useful, and I saw Iran here. And then using our whole cell model secret sauce, we're able to actually start generating simulations that are able to come eerily close to what actually is going on in a real cell. And I'll just show you one version of this. So here's an E. coli cell growing. This is all data coming from the simulation. What we currently have, we call the half cell model or the 2/3 cell model, but it does grow repeatedly over many generations, and it can grow in multiple environments, and it can actually, as it grows in these environments, sensors will be activated, gene expression will change, metabolism will shift, number of ribosomes will go up, the DNA will start kind of parallel processing there with multiple replication forks, the cells will grow faster and get bigger, and we can see all of that and much more in kind of one unified simulation, which honestly we are still trying to figure out. We've done a number of fun studies with the model, some that I'm just posting here, looking at operon structure in the model, how growth rate control impacts growth and what it depends on, and even discovering some new regulatory aspects, which I will actually show you in more detail today. And what it really comes down to is that I feel like we're hitting this new stride where we're able to do things much more difficult than what we did before, which I'm super excited about. So here's a hierarchy of my needs. So maybe some of you know Maslow's hierarchy of human needs, belonging, love, these kinds of things. As an engineer, I lack the ability to functionally engage with people, so I have a totally different set of needs in my hierarchy. Just kidding, obviously, and sorry to all the engineers out there. But when I was just starting out as a graduate student, the biggest dream I could possibly imagine was to take my model, leave out some of the data that I was putting in, and see if I could predict, those data as a validation of what I did. And I remember still the first time I ever did that. It was incredible. It's really what got me started in the field. And now this model, just because of the sheer number of data and the sheer volume of what it purports to do, we're able to compare it to data in a way that really has never happened before in biology. And so that validation step is actually very extensive and exciting still, but it prompted us to go up the pyramid. The next thing was, well, could we make a prediction before any experiment was actually done, right? And I remember, again, the first time we ever did that in the whole cell model many years ago now, in mycoplasma, and I thought that that was the coolest thing that had ever happened to me, basically. And but now in this E. coli model, we have many more chances to compare it to data, and we've done this kinetic parameters, we've done it with decay rates, we've done it with different other things, tRNA stuff. So we're kind of moving past that as well. And so we're starting to really look at the top of the pyramid. And the top of the pyramid is, what new paradigms are there in biology that are out there remaining to be discovered? And the way that I try to explain it simply to my lab is just to say, CRISPR was found in E. coli. There are at least 10 more, Nobel Prize deserving big discoveries that could still be happening in these simple cells. And even though, prize hunting is not really our thing, but the idea of looking for large fundamental things that really fundamentally change how we think about how cells behave, that's the payoff that I want from these models now. And so hopefully you see what I mean when I say like a transition point here. I'm going to show you a couple stories. The first one is very much kind of data focused. The second one is just to show you how far down the rabbit hole you can go with this model. And the third one is just this brand new stuff that I couldn't resist. You're going to get a little bit of concentrated orange juice today because I'm going to try to go fast to cover everything. But I couldn't resist showing the latest stuff we've been doing with metals. So first, let's talk a little bit about subgenerational gene expression. And this was really a discovery from some of the earliest sims we made with the E. coli model, and it has to do with how things get expressed and what their dynamics look like. These days, with all the sequencing, et cetera, we don't get a chance to focus on dynamics enough. I'm sure I'm preaching to the choir here, but thinking about dynamics can really give you a lot of interesting insights. And so when we looked at the dynamics of how genes were expressed in these sims, we found something very surprising. Specifically, if you look at the upper left, okay, we have cells for all of these. We have cells growing on over 6 generations. And we have 2 different genes, this one that's labeled exponential, RNB, and the one that's labeled subgenerational, CCMB. The one on the left, let's look at it for a second. This is what I expected to see, and this is what I saw, right? So basically, there's a lot of stochasticity kind of built into the model, but over these 6 generations, the amount of protein kind of goes up, and then the cell divides, so it goes down by half, and then it kind of goes up again as the protein is produced, and then it divides in half again, and it goes up. So that has an mRNA dynamic that's associated with it. There's more randomness there because of the short half-life of the mRNA. You can really see it. So there's a lot of kind of going up and down of the mRNA, but that mRNA is directly being translated to produce the protein at the top. Like I said, that's what I expected to see all the time. And that's what I did see a lot of the time. But I was shocked to see this thing on the left. So what you have here, look at the bottom right for a minute. What you have here is a gene in which only one mRNA is produced over 6 generations of time. And when that mRNA is present in the cell, a lot of protein is made, like 150 proteins are made. And the protein decay rate is very slow, very low. So as a result, every time the cell divides, it goes from 150 to 75, and then you have another cell cycle. And then when it divides, it goes back down to like 38 or 40, back down to 20. And so incredibly different dynamics, right? Incredibly different dynamics. And what really shocked me about this, if you look at all of the genes together ordered by their synthesis probability, subgenerational gene expression is not the exception, it is the rule. So about a third of the genes have that exponential characteristic to their dynamics that I showed you before. Close to 60% are actually not being expressed every generation. And so you have this really interesting and unusual dynamic that shows up. Why would this be the case? Well, it actually has a really interesting phenotypical, like it explains an interesting phenotype, which is to say that, E. coli has to respond to a lot of different environments, some of them positive, some negative, right? On the positive side, maybe you get an extra carbon source. On the negative side, maybe there's something that's going to damage the cell. And, you've maybe seen different people talk about E. coli as being like a motherboard or a computer circuit or whatever. These days, I think about E. coli as a suitcase. It can't hold everything. It can't hold every protein that it needs. It's not ready for every vacation it could possibly go on. There's no room for a surfboard and skis, right? It needs to be able to somehow, though, as a group, respond to all these different environments. And so instead of every cell being ready for everything, which is kind of how we would think about it as humans, in E. coli, every cell is ready for something, and it's the colony that's ready for everything. So what you see here is if there's a shift to a lactose media, then those couple of cells that have the lactose uptake machinery will outgrow the colony. They will establish some control to keep that thing constant, and then more diversity, phenotypic diversity, will reestablish itself in that community, if that makes sense. So that's what subgenerational expression could potentially explain. But the question is, does it actually happen? And so what you're looking at, these top three rows, we have our simulated expression of 3 subgenerational genes at the left and then 1 control at the right. And here there's a whole bunch of generations that we've put together. So if you look at the HUP-A gene, again, first 3 rows are simulation. What you can see is that they are being expressed exponentially. They're always being expressed. And actually, when we go in and make a reporter, in chromosome reporter of the HUP-A gene and its expression, you can see, and it's just a simple fluorescent protein fusion. I say simple, but anyone in the lab who works in the wet lab knows these can, especially in chromosome, can be very difficult. But what you see in that colony of E. coli is that all of the cells are expressing it, and they're all expressing it at essentially the same level. Whereas if you look at these 3 on the left, this is what the model would predict. Subgenerational expression means that there are islands of expression where a single cell has had a burst of expression and its daughters, its children, are roughly half around it and then roughly half as it goes out. And you can see exactly that subgenerational pattern, the dynamics in that 3rd row, right? It goes up, divides in half, it goes up again. We can even see this kind of amazingly in single cells using the mother machine developed by June. So at the bottom, I've just labeled the cells with a fluorescent plasmid. It's the same, looking at it a different channel on the top. You can see these little flickers. If you pay attention, you can see these flickers, and some of them are bigger, like there was a good one. And there's a big one coming out kind of right in the middle. There it is, right? So you can actually see subgenerational gene expression happening essentially in, almost in real time here, which is really exciting. And when you quantify those images, sorry, it's a little grainy here on the left, but when you quantify those images, you can see what our predictions look like, that they should have this kind of special dynamics that looks very different from exponential expression. And then when you take those actual images that we generated and look at the quantification of those images, you could see just this beautiful parallel between what the experimental data is doing. So this is kind of my idea of a paradigm shift, like thinking about the expression in cells, thinking in more detail, and then going in, instead of looking for a parameter, which we certainly also do, looking more for a conceptual validation of a new phenomenon that really hadn't been appreciated at the same level before. Okay, that's number 1. Here we go with number 2. Number 2, again, like I said, I want you just show you how crazy the simulations can be and how much they have to show. But again, it will come down to a new paradigm. So we were trying to do something related to tRNA charging, which previously had been kind of a weakness of the model. And as we started looking in how to improve it, we started to realize that there actually was a pretty significant controversy in the literature, a 40-year-old controversy that had not been resolved. In particular, it's Jakubowski and Goldman had gone in and they had looked at all of the measured K-cats for all of the tRNA synthetases in E. coli, and they had compared those K-cats with what the demand would have been known to be for just the protein content required for a cell to divide. And what they found was pretty shocking, and it kind of goes back to something we all have thought about at one point or another. I think for me, I used to just consider this a gotcha question when I would do my talk, but I now realize how important it is. It basically has to do with in vitro measurements being extrapolated to in vivo, predictions, right? What do we know from an in vitro estimate? And what they said here is that the in vivo activity for almost all of the synthetases they considered is orders of magnitude higher in vivo. The in vivo activity that's required is orders of magnitudes higher than what was measured in vitro. Not much was done, no one's doing the in vivo measurements of these things, right? And so that not much could be done over these 40 years, but as we looked at all of that, we thought we could actually add something to the discussion because we can simulate the in vivo context, right? So that's kind of an interesting thing you can do. You can simulate the in vivo context and then plop in those parameters and see if indeed they are sufficient for the cells to grow. And if not, why not? So I'm going to whip through this stuff, but we took a relatively simple model, made it much more complicated. Hee-Jo Choi was amazing in this. And then she put in all of the kind of high measured in vitro determined synthetase K-cats. And sure enough, what she found is that the cell was not able to grow at its expected doubling time rate. It was like threefold or more increase in that doubling time. And so we had a clear indicator here that Jakubowski and Goldman were right, that these K-cats were not sufficient in the in vivo context. And we could further prove that to ourselves by going and looking at the flux in detail, assuming everything was at its highest value so that we've just looked at K-cat and enzyme. Then because the model simulates so many things, we could go look in detail at the enzyme count predictions we were making and compare it to an actual measured proteome. As you see, there's very tight measurements. So we realized it was indeed the K-cats that were problematic. And even at a single cell level, the distributions that we were simulating looked a lot like the synthetase enzyme distributions in single cells. Don't worry too much about these. This is just us trying to get confidence that we were kind of going after the right thing. And here are a number of the other comparisons we made just to make sure we were on the right track. Maybe one point I'd note here is these models are, at this point, very intensely monitored and validated to the extent that we can. Here's the money slide from this talk. So we took all of the synthetases and we just compared them. The gray is the range of in vitro measurements. The blue is our prediction for what the in vivo range should be. And you can see if you're at the left, there is some overlap. As you go towards the right, there's more and more of a difference. I'll just focus on the ARG-RS, the arginine amino, the tRNA synthetase for today, so that with the green box around it. Because what we can do then is we can do this kind of fun counterfactual. We can actually just see what happens when we put, what we predict the value should be against what the high in vitro measurement is. And so the gray bar there is the control, right? This is how fast it should be doubling every generation, more or less. And at the right is what happened when we plopped in those other that other parameter. And you can see right away the doubling time jumps up and it keeps going up until it hits, for us, the checkpoint where we automatically cut off simulations. And also the failure rate increases. That was interesting to see, but it's more interesting because we can actually go in here and start doing some detective work, some diagnostics. The green bars that you're looking at there basically tell us when production of something flatlines. So you can see that if you look at the bottom, the total dry, you can see that the total dry mass flatlined at generation 12. RNA stopped at about the same time. DNA stopped one generation earlier. Protein stopped at generation 9. So we thought, okay, that makes sense for us to pursue and see why it is flatlining. It also made some intuitive sense because we're talking about tRNAs, we're talking about translation. So that made sense. So sure enough, we thought, well, let's just see if all the tRNAs are being amino acylated. Maybe it's arginine, maybe it's something else as well. Turns out all the tRNAs are getting amino acylated, with the exception of arginine. But the big deal here, and here's where I have these gigantic arrows for my Zoom talks, I apologize. But here we have a much bigger deficiency in arginine than would have been predicted by the change that we implemented. So something weird was going on here. Clearly, it goes almost to zero, and we weren't sure why. So we can just look at all the inputs of that reaction, the tRNA synthetase reaction for arginine. And what you see is the synthetase is essentially the same, whether you use the in vitro or in vivo measurements. The arginine tRNAs are about the same. ATP is about the same, but big arrow alerts, the amount of arginine as a substrate is way down. That was a shock. Like, we did not include anything like that in this model. It's not clear why it would have been in there, But why would the arginine substrate be the problem if you've reduced the KCAT on the synthetase? Well, for that, we can keep going down this rabbit hole, and we can look at all the metabolic fluxes that kind of culminate in producing arginine. And what you see if you look at the right, sure enough, at generation 9, arginine is out, and then the model kind of goes off into self-destruct. But at the beginning of the arginine biosynthesis pathway at glutamate on the left, it's making glutamate just fine. And if you go through all these steps, what you find is that the N-acetyl-glutamate synthase, ARG-A, encoded by ARG-A, is tanked out at generation 9. No reaction flux is being produced, and the enzyme is gone for some reason. Big arrow. So you can go look at the expression. You can see that the monomer is basically not being produced. It doesn't seem to be a problem with degradation, but there is no hexamer. And then we did an experiment. This is kind of fun, ribosome profiling. It can be a challenge. It's a relatively new and somewhat challenging experiment to perform, but we can do it in silico very easily. And so if you're looking at the left, you'll see the in vitro or the in vivo estimate of the KCAT that we have. And you see there's basically no stalling on the sequence, right? Looking at the ribosome stalling. There's almost no stalling, and it doesn't matter. Importantly, it's relatively even amount of stalling everywhere in the sequence. However, using that in vitro KCAT, there is a ton of stalling, and almost all of it, if you just look up, follow that tall green peak up, and you'll see CGG. Remember those letters. 90% of the time, ribosomes are stalling on CGG in the sequence, which was, again, like to me, this whole thing was just amazing. So then we looked at the transcript, we looked where the stalls were happening, and it turned out most of them are happening with a premature ribosome termination at this CGG-CGG tandem repeat. Okay, and that really struck me because here at Stanford, Charlie Yanofsky, many decades ago, discovered something in E. coli called attenuation, which is a form of translational regulation that importantly depends on dual repeated codons, as you see here for tryptophan. So all of a sudden I was thinking, wow, what's going on with these dual repeats? And so we went, long story short, we found all the cases where there were proteins that had the dual repeat, and we showed that indeed like 20-fold reduction in expression from those sequences. And then our reviewer had the ingenious idea of, because I hadn't really, I'm still kind of learning how to think about this as like a real bug, but they said, well, if this is a real bug, you would replace CGG with a more common codon. So what happens if you do that? So we replaced CGG with CGU, and sure enough, it rescued the phenotype, right? So that was really interesting. And so what I want to tell you here, I mean, bear in mind, the crazy thing is I've just told you this story. Not a single bit of data, new data was generated, right? This is completely just all the craziness that you can see in a simulation of a cell, a kind of a digital twin of the whole thing as it goes over time. And you can look and you can see all these tRNA rates. You can see exactly where the stalling happens. You can see how this impacts expression of the hexamer. You can see what that does to the fluxes. You can see what that means in terms of arginine in the cell, yada, yada, yada. And all of a sudden you're all the way up to cell mass, right? So it's pretty incredible to see just how much you can consider. And we call this process deep curation. In the end, it turns out to be a positive feedback loop, and we're starting to pursue this experimentally right now. It is a little challenging, but people have seen that there was, once we knew what we were looking for, we found a really interesting thing where someone was studying HSV2, but they were looking in particular at these dual rare codons. And there's this other really interesting aspect that you have both positive and negative feedback impinging on expression of ARG-A. The negative had been known for a long time, but not the positive. So we're excited about that. But here's my very favorite story because, it's always like the baby in the family, right? This is my newest story. I'm excited about it. Here's Cyrus Knudsen, who did tons of great work here looking at the metals in the cell. We haven't published this. It's brand new. Looking at the metals, it's interesting because metal has so much relevance, right? In industry, bioprocesses, in immunity, where we're constantly so there we go. I said the word immunity, so this counts. So there's an immune, as we're always fighting with bacteria for the metals like iron and other things. And of course, even in the ocean, there's a lot of metal limitation, right? As if we were to dump iron into the ocean, we would see a beautiful bloom in some places of the world. And so we wanted to see how to incorporate, because we're always trying to incorporate every single thing we can include. And Cyrus was like, look, there's thousands of experimental metal cofactor interactions that have been determined. So why don't we put that together and see if we can generate for the first time a simulated metalloproteome of a single cell? I'll go through this really quickly, but just what the data kind of looks like, there are some enzymes that hold up to 96, different metal ions, right? And some that only have one, or some that might have a mixture of several. This is about a fourth of the total, but including all of that was what we were trying to do here. And when we did that, hey, Cindy, when we did that, what we found was very surprising, actually. The model was, I was not expecting this kind of validation. It led us to do a little bit more here than we normally would. But when we actually saw if including this enabled us to predict metal counts in bulk and compare them to experiments, we found that we were extraordinarily good at doing that, right? So it made us, and of course there are exceptions, like look at copper, for example, but it was surprisingly good. So it made us think, wow, we should look at this in a little more detail and see what we can learn from these simulations. And it was even more interesting when we started looking at the single cell distributions, because here it is for iron in single cells. Those might not look completely overlapping to you, but for me, especially when you consider that these are attograms we were talking about, right? That's a pretty significant. We didn't use any of the experimental data for either of these two comparisons that I'm showing you. So we went forward and made our own prediction of where all the metal ions are going in the cell under a condition here, right? Like under a single environmental condition. And what you can see is exactly that. We have iron, zinc, manganese, copper, and then there's up at the top, there's a tiny wedge with several other metals in it. But you can see by functional category and by gene, or by protein really, where everything is going. So I'm just going to tell you two quick things here. I'm cognizant of the time. First is for zinc. So we were looking at zinc, like the metalloproteome for zinc. And here you can see kind of where everything's going. And really what jumped out at us that was kind of this crazy thing is that the top two seemed like they could be competing with each other in some way. You see that 50S ribosomal component that contributes to growth. Methionine biosynthesis also contributes to growth in the absence of methionine. And so that was kind of interesting that they were both such big demand proteins for zinc. And so we had this crazy idea that they might be competing. And here is my favorite experiment we've done a long time because it was simple, it was cheap, and it has told a big story. So this is Adrian's work. He basically just grew E. coli in a defined rich medium and found growth rates. And you can see the 3 data points in the average here, right? And so now we're looking relative to that. He then took methionine out of the medium, okay? And he got a reduction in the growth rate, okay? I don't think anyone would be surprised at this part. What's really surprising is that he then added zinc back into the medium, not a carbon source, not something else. He added zinc back into the medium and rescued that growth rate, right? Meaning that there was a trade-off and a competition in here predicted by the model that actually has an impact on growth. This got us really excited. So then we went to a colleague, Max Saito at Hui, that I've been working with together with Iran and others as part of this group called CComp. And he can do metalloproteome experimentally. It still has, I wouldn't say it's as quantitative as what we're predicting, but it was enough that we could start looking in detail and seeing if our predictions were correct. And so I'll just show you here. like in this plate, it's showing where all the zinc is on, think of this as kind of a 2D gel of protein separation and then seeing where the zinc is in that separated gel. Now I'm going to turn it so you can look at it from the top as just a big heat map. And there are two big peaks as you see. And sure enough, this one is the methionine biosynthesis protein, and this one is the ribosomal subunit, right? So pretty amazing to see how this is going. And we're kind of still seeing what will happen, but I'll tell you one last little thing. This gave us some excitement about thinking about growth, manipulating growth rate with metals. You know, typically when people culture bacteria, they're really using just the trace metals that are just available to them and not something more well-defined than that, or at least that's often the case. And so molybdenum was interesting because when we looked at the metalloproteome simulated for molybdenum, there was really one enzyme that dominates. It's this nitrate reductase, which is used anaerobically. And so just to share kind of this brief thing, we found that it was really expressed primarily under anaerobic conditions, which gave us this idea maybe we could make a faster growing E. coli under anaerobic conditions just by adding molybdenum. So we went and tried, you know, because too much metal is toxic, too little is not enough. So we did a little bit of an optimization here, but in the end, we were able to get an anaerobic culture that is growing much faster than base media and almost at aerobic rates just by adding molybdenum, which is the kind of thing that I think does matter in terms of a biotech process, right? Or somebody who wants to get cells in a fermentative process to grow faster. That means a lot in terms of a potential industrial impact. So we've been super fortunate to have funding from the Sloan Foundation to try to get 90% of the annotated genes in about 5 years. I'd say we're making good progress, but it's been fun also they indulge our diversions like this metals one, so that's been great. And I'm just going to close with one thing because it's especially these days with David Goodsell retiring and David Goodsell partnered with us at some point. One of the questions I get very frequently has to do with including proteins. This is for Mycoplasma genitalium, but as a huge tribute and appreciation for everything David Goodsell has done for me and for the entire community, the scientific community. We worked, he actually took some of our whole cell simulations as a baseline, and this was Martina Mariton and some other great people. So he was able to take those and add in the structures of all the proteins in there and able to actually make these interactive 3D models, which are pretty incredible. They're not dynamic, but more recently, this guy in Canada, a software engineer, Timothy Davison, actually took that and turned it into an app on the Apple Vision Pro. I think you now can get it on the iPad or iPhone, but it's amazing in the Vision Pro, where he actually takes these same models and he makes these interactive, explorable 3D representations that, again, are kind of based on this whole cell modeling framework, but then takes all of their amazing cell pack and structures that we've generated. So I'm just going to close with that. Here's the lab. We rented a bunch of these for a Stanford cell tour day where we had lots of people come and tour the cell virtually. I think it's so amazing to be able to look at these things in their holistic context. And I just wanted to shout out to David Goodsell, who's inspired me in a million ways. Many thanks to all these incredible groups that have taken a chance on a long shot to fund us, and happy to take any questions now. Thanks for your attention.
Okay, thank you for the great material. Let's open the floor to questions. Tom, did you have one?
Bill has his hand up.
Oh, great.
Oh, sorry, Bill, go ahead, please.
Oh, yeah, hi. I was wondering if you have any thought of moving towards bacterial behavior, chemotaxis, maybe at first not even having the thing move. Well, the virtual world could move around it rather than have it move and worrying about flagella and things. It's just in terms of tumbling versus straightforward movement.
Yeah, I love it. So actually, right here in the audience is someone who could give us another 45 minutes on this topic. Eran Agman, he's now a new professor in the University of Connecticut, and he developed a software framework, Vivarium, which enables us to do this more multi-scale type stuff, where we have multiple cells interacting. Each one is an instantiation of the whole cell model, and they interact together in a shared environment. Iran has also worked with chemotaxis. We haven't quite engaged that into the full framework where you're just seeing them all swimming around each other, but they do push each other around and grow and stuff like that. So it's definitely very much on my radar. I want to go from, we've done a little bit of like from whole cell to whole colony models. But I really think that's the future. Very exciting. Yeah, thanks. Great question.
Other questions?
I see something for Eric Moody. Could I show the slide citing the page? Sure. Okay. Let me find it. It is there. You got it? Okay. See some more. Yeah, okay, got it. Anything else? Liliana?
Yeah, hi, can you hear me?
Yeah.
Really interesting talk. I'm here at NIID, and we have a lot of issues with antibiotic tolerance and heteroresistance and kind of these phenotypes that we can't seem to explain simply by gene expression. I'm wondering whether you've had any thoughts on this and explored that question at all.
So do you mean in bacteria?
Correct, yeah.
Okay. And so sorry, just give me a little more clarity. So
Okay, so the
gene
antibiotic tolerance is basically a
Mm,
phenotype after you've exposed a
yeah.
colony to a very
Yeah.
high concentrations of drugs, and then the survivors have this tolerance, not resistance, right? So you look
Yes.
genetically, they're not resistant, they're just tolerant to the antibiotic.
That's right. So I see that as a subgenerational gene expression phenomenon. So that's how I interpret that. And interestingly, recently we've been partnering in a joint project that I'm hoping we will finally get across the final clearance finish line with Iran and with Min-Soo Kim at Emory, who's an antibiotic expert, and with some other people, Suk-Jun Jun. This hopefully funded by DARPA to go after antibiotic responses and their heterogeneity in the context of that low expression. Yeah, those low, rare expressed genes. Great question.
Yeah, super interesting. And just really interested to see how the models how we can do these simulations.
Yeah.
And how they may help us develop better antibiotic courses or even antibiotic drugs themselves.
There is a paper, which I'm sure Iran will put on the chat, which basically shows some simulations of how this works with antibiotics in colonies based on whole cell modeling. Thanks, Iran. Tom.
No, fantastic talk. Thank you so much. How do you I'm assuming you're also working with the biotech industry to translate these the way you can optimize the generation of the cells how is that working on the translational side?
Yeah, that's a great question. I think, okay, so, that's one reason why I said I think we're hitting this transition point, because there actually have been several companies and entities who've tried to start something related to whole cell modeling. And I think it's always been a little premature. So some of the efforts out there, like Google X hired a bunch of people out of my lab. There's another company, kind of a hedge company called Deep Origin that hired a bunch of people. And I think the ambition was really high, but it is hard to do this, right? I hid a slide in which I talked about like what makes like an AI or LLM driven virtual cell desirable as compared to this, or how does this outperform that? You know, there are advantages to each. I think the best argument for like an LLM or AI driven thing is that it's just hard to do this. Like, you know, this is a very intensive, detailed thing, and I think we're getting better and better at it. But, you know, a lot of the LLM stuff, maybe it won't be as valuable, but you could do it right now. So it's kind of, although it has its own problems with the amount of energy it takes, you know, there's definitely several advantages of using this, which I'm happy to talk about. But now, and in fact, this is what DARPA, maybe just to finish the thought really quickly, what DARPA wants Iran and me and Minsu to do in particular is demonstrate real translative capacity in two areas, three really. One is for a small molecule that we're still negotiating on, we have to show that the whole cell model can predict alterations to a strain that actually lead to a physical improvement in production of that strain. That's an extremely hard challenge, but they want to see that in 18 months. The second thing that they want to see in 18 months is that we are able to generate an MIC, a killing curve for an antibiotic that has not been determined, and then show that we were able to get it or not get it experimentally in 18 months. And the third thing is that we have to have our demonstration capacity has to be so advanced that someone at DARPA could independently validate and show that we did it. It's not just like something that we said we did from our lab. If we're able to succeed at that, which I am highly believe we can do, but it sounds super hard. But if we're able to succeed at that, then that opens the floodgates, I think, for translative ability in this space. So I'm thrilled that DARPA is doing it. I'm frankly very, very nervous, but I'm nervous in a good way. I'm excited.
Amazing. Congratulations. Thank you.
Yeah, thanks. Hey, Steve.
Hey, any spatial modeling interest here? Also, would space add anything to your model, do you think?
Oh, great. That second one is a doozy, right? Okay. So we think about spatial modeling a lot. So we have kind of this higher order colony-based spatial modeling, you know, where we're modeling, you know, I think it will get more and more sophisticated. But I'm guessing what you're asking is more like, what would this be if it was an all protein or all molecule or all atom simulation like some others do?
Right.
I think there's a ton you can learn from those kinds of things. What they're typically missing is function, right? So I can show you that big, beautiful picture of all the proteins, but what you will not see is any of them catalyzing anything, right? So that's kind of the complexity there. And we tend to be very function heavy. So I see it as very compatible, but not quite. So now I'm going to ask, can you see this whiteboard?
I see it. I can't see what's on it.
Oh, you can't see what's on it. Darn. Even as big as you can make it. Okay, I was gonna show you. There's a cell up there that I drew. And basically the idea is, I think after having consulted with many different groups who've done a lot of protein localization studies in E. coli, I think that there's a middle ground for us to stake out. Like right now, the current model has maybe 4 or 5 compartments, like the interior, the inner membrane, the periplasm, the outer membrane, the outer environment. But I think there's room to say left pole, right pole, nucleoid. And I think those areas will have different expression of like, or different localization, like the ribosomes and the RNA polymerases will be mostly in that middle, which means that they will be depleted in the poles, which will further be enriched for, let's say, the glycolysis chain or whatever, right? And if you think about DNA as a big thing of chicken wire, you can kind of calculate the holes in it. And it turns out there are a lot of things that do not move through those holes, but ribosomes can fit in there. So anyway, all that is to say, we're definitely thinking a lot about it for the cases where I think that it will impact function in particular, function and phenotype.
Oh, thank you.
Yeah, thank you. Ellen.
Hi, very great talk. I've worked in bacterial cell division and such for decades, and so
Okay.
I was listening to everything that you've been talking about and just going, oh my God, there's so much you could do. So one of the questions though is cell division in E. coli has been extremely well studied. And I think, that would be, if you could recapitulate even just the cell division cycle and everything that's known about that, that feeds directly into all the antibiotic resistance and antibiotic, you know, um, part that you were talking about, basically. So the other thing is depending upon whether you're thinking about these models from what does E. coli do when it's aerobic and in defined rich media versus minimal media kind of thing, because the metabolism is different, right? And things that affect it when it's in these wonderful aerobic rich, environments versus, how they respond when they're in minimal media and how they use amino acids differently too.
Yeah.
Right. All of these things impact the metabolism, impact the transcription and translation, impact cell division, impact how antibiotics are either used or not used, like kill or not kill, because when they're in minimal media, they grow a little slower. So you have to use much greater quantities of antibiotics to do the job. And you can also, when they're in that situation, that can also cause antibiotic resistance to develop, and, or they have mutations that can happen that, prevent them from even uptaking antibiotics. And so that the MIC also would be quite different in those different kind of media. And so anyway, not sure what my question was exactly, but I guess, have you considered actually doing these models based on cell division and thinking about just that as the focus? Because that, if you study all of the cell division and DNA replication that undergoes during those different media shifts and things, you will get to your antibiotic resistance problem, I think, easier.
A lot of great questions in there. And I think the short answer is I would love to learn more from you, like pick your brain, basically everything you know about cell division. We do have cell division included. It's more on the, it's not qualitative, but it's more on the qualitative side. It's less descriptive, less mechanistic than I would hope that it would be. So we're always kind of trying to build it up. We definitely do see interactions between metabolism, cell division, et cetera. And one fun thing is that exactly what you said, we pitched this to DARPA that we were going to take an antibiotic where the killing curve was known in one condition, but we were going to predict it in an unknown condition, right? So that was would give us a way to kind of do the parameter estimation, make sure we're on the right track with some validation, but then go into something where we would predict a much different MIC curve and then go show that we were able to get that. I also think maybe this is another fun unpublished thing, but we've been noticing, surprisingly, we've been studying a phenomenon when we just overexpress genes in E. coli, like in the Sims, and what makes them grow more slowly, what makes them shut down. And I've been surprised how often copy number starts to show up. And so that was, anyway, a big surprise that we're kind of just currently looking at.
Yeah, I should really set up a meeting with you and Aaron, because there's so much opportunity.
Yeah, that'd be fun. Mm-hmm.
Thank you.
Yeah, thank you.
Other questions? And I was struck at the beginning, you talked about this distinction between genes like that, where you have continual synthesis and situations where you have burst synthesis followed by dilution on growth and division. And I remember visiting Stanford, and I'm trying to remember who I was talking to who was doing statistics. Maybe it was was too long ago for it to have been your group, I think, where they were showing that stochastic choice of differentiation state, for example, was the best you could do because the bacteria didn't have enough information to make an informed choice about which pathway to pick. And so the best you could do was pick randomly among a variety of options, each one of which had some survival benefit under different conditions. Then the question was how you randomize to get the appropriate weighting of the different conditions. So that's one direction that part-time selection that you talked about, where you have the backups, goes. The other one, though, is in multicellular organisms, where you have to control the size of organs and have the turnoff of growth at a particular time. And they'll also use the mechanism where you have synthesis up to a particular point in development, and then you'll have dilution, where the dilution then acts as a switch to turn off growth. And people like Langer have talked a lot about that as a mechanism for size control. And so in the case of the bacteria, if you perhaps the first one is the only one that's significant, but you could imagine that in colony formation, there might be collective regulation mechanisms of size that could also work the way your dilution mechanism was described.
Yeah, interesting idea. I hadn't thought about it. Yeah. So right, that's what's going to keep me up tonight. It's a good idea. I think one of the things that's kind of interesting with that dilution, right, is it really is fairly limited, I would say, just to how often an RNA polymerase is making it to that promoter, because the protein degradation time does not change appreciably. Like they're much longer, but it essentially, for all but a small handful of proteins, they're pretty much governed by the dilution rate, right? Every time they divide, that's really their big loss. And then the mRNA half-life doesn't really vary much between, 3 to 6 minutes, right? So there's not a lot of leeway there. And translation is kind of set, the rate of translation and the rate of transcription once it occurs is set. So a lot of it just has to do with the initiation of transcription that really depends. So I think of it as differentiation on the cheap. You're just basically regulating affinity. And for all of that, you get a pretty good, it's also really interesting to think about how E. coli is basically making use of, I think when I was starting as a graduate student, we thought a lot about homeostasis and how cells maintain robustness in the face of noise. But I was really excited when I kind of started here, people started thinking more about cells generating their own noisy conditions for important phenotypic reasons. And that's what you see here. That's what you can also see in differentiation of the fly eye. There's been some great work, even in my old, kind of first love of NF-kappa B and innate immune signaling. There's a certain amount of this. And I think it's worth being its own broad new field, having its own conferences, just the idea of not just robustness, but just actively generating noise, exploiting noise for phenotypic reasons.
Right. Well, certainly in the case of trying to ensure that you have all of the possible survival strategies represented, then the noise, if it's going to be effective, the noise has to be regulated, because you have to ensure that you have the appropriate distribution of outcomes.
Right. And here's the killer headline: the noise has to be robust.
Yes, also.
Right.
I mean, of course, there was all that discussion in the past about under harsh conditions whether the rate of mutation goes up because you have stress, and and that's degrading the accuracy of transcription and translation, or whether there's actually active regulation, which is increasing the rate of mutation.
Yeah.
So,
Yeah. Fascinating. I see one more question down here from Hayden. Any quick thoughts on why the Apple Vision Pro? So I had no input. I had no, this is just the miracle of our current world. Someone just saw this, you know, a computer software guy in Canada saw this and decided to do it. And now it's been done. It's one of the top education apps on the Vision Pro. So we're just, so I've just been rolling with it, which has been great.
Yeah, great. Thank you. I was just curious if there was any particular motivation or if it was just who and what was available. So,
Now, maybe let's put it in a historical context. I mean, one of my favorite things about, two of my favorite things about the Genome Project, right? In both cases, first you had Gene Myers come up with this idea for shotgun sequencing and essentially launched him. Venter gets a ton of the credit with some justification, but really Gene Myers launched the Cellular thing way ahead of the public effort just by having this idea of shotgun sequencing. And then once they were all making everything public, and then a random, I think, graduate student was the first to assemble the human genome just because he saw it and he thought I could do that. Everything's public. And so he just did it and he beat everybody. And I think that's the real fun about making things public. Maybe not fun for the public effort, right? But fun for everybody else. Like for people who want to these days, you can come in and you can crash somebody's wave in ways that Francis Crick only used to dream of. You know, everybody thought of him as this horrible pest, but we could do that at scale these days. I've never done it to anyone, but it's astonishing when it happens, and I personally love it. I never would have thought of that. So having Tim do it is special, you know, we all win. Okay, Boyan, the dynamical consistency of the overall seems spectacular, right? What do you foresee as next steps towards improving? Yeah, you know, it is pretty fascinating. We tend to use these parameters just out of the box, so we try not to change them. So in the first publication, I think we probably fit, you know, out of 20,000 or 19,000 parameters, I think we maybe fit 50 something in there. But what it means, just to be clear, is that we have to make, because of course they're not all internally consistent. And in fact, that was the whole point of that science paper, was to show that they're not. But we had to come up, we were kind of banging our heads around trying to make a purely ODE based model, and that was never really going to work. So we had to come up with methods, and you'll see there's a lot of optimization in the simulations. Like instead of representing something as a set of ODEs, we could represent it as an optimization problem that's minimizing the distance from having those ODEs be correct. So there's kind of a lot of oil to kind of lubricate the works so that these parameters play nicely against with each other, and also so that we can identify the ones that have the worst match. So there is some kind of fun, you know, we talk about the math, we talk about the experiments, but the software engineering and how that all comes together, the compute, you know, the kind of numerical recipes component is important. What do you foresee as next steps towards improving? Yeah. You know, Michael Stumpf, who's was in London, but now in Australia, in Melbourne. He wrote a paper called, I don't remember exactly what it's called, but something like parameter is, I mean, the top line was estimating parameters in a whole cell model is a disaster. It's true, right? I think this is a space kind of like weather prediction, you know, had people thinking about chaos theory, right? Which they never had to think about before, but it ended up really benefiting a lot of other fields. Or they do this incredible data assimilation, you know, that benefits a lot of other fields. I wonder constantly, what are the new mathematics, statistics, hardware innovations, software innovations that just trying to do a massive project like this will enable, or at least inspire someone to think about. Maybe I'll tell you one more funny thing. When we first did the first model, we had a reviewer, you know, that the famous reviewer 3, but I think even reviewer 3, right? They might not tell you what you want to hear, but they, you know, it's like Disneyland. You learn a lot more about who you are in line than on the ride. Right? Like you learn more from these hard reviewers than you learn from the easy ones. So we got a hard reviewer who had some really interesting questions, but one of their things still haunts me to this day. We had said we had validated the model and they disputed that we had validated the model because they said validation only works with independent data. And because the model has everything included, as you claim, there's no way for anything to be independent. Therefore, it can't possibly be validated. Therefore, you did not validate it, right? And that is a fascinating argument, right? In some ways, we had modeled ourselves into a validation impossible corner. As an engineer, I'm comfortable with that. You know, maybe a mathematician is out there, you know, carving themselves up into 7 Horcruxes over that. But I think but for us, you know, we're just going to do our best to say, okay, we're building confidence by the comparisons we make, recognizing though that it's really, you know, intense. So I see Pedro, I hate this corner and I'm on it right now. So right. So it can be really tough to find yourselves in these spaces, but the good news about that is huge opportunities for anybody who's thinking about in those directions. You know, we would jump right on that. You know, I listed all those. I could list another one, like how do we make causality theory hyperdimensional? You know, you could because this model is like an infinite source of counterfactuals, right? So how do you get into that? Yeah, there's a million things to wonder about at this point.
I think your point about data assimilation is well taken. There was an earlier question about relationship between these kind of mechanistic models and deep learning models. And
Yeah.
one of the big advances in deep learning is the ability to rapidly correct the models based on user input, for errors. And so, how to maximize the effect of of recognized errors or anomaly detection, and use that to update models. I think that the machine learning is in a sense more advanced than mechanistic modeling in terms of approaches to data assimilation at this point.
Yeah.
And I'm not sure how we can do better in the mechanistic side, and take advantage of that.
Yeah.
I wonder about your ideas on that.
Yeah. No, that's really interesting. And maybe this is a good segue for me to show the slide that I originally skipped. So here's a thing that I wrote for the DOE, where they're thinking a lot about this kind of stuff right now. And I just highlighted a couple things. Here are, in yellow, I've highlighted what I think are the advantages of a mechanistic system. One being the explicit, I think everyone would get that, the explicit embedding of causality, which can go all kinds of interesting directions in terms of scale. As a result, the fact that the model predictions are explainable, right? That unlike, you know, again, you guys will probably already know this. There's also this kind of interesting thing that the mechanistic models learn in a totally different way than the neural network-based architectures. And I think that's really important in a couple of ways. One is less data and more targeted data. And one relates to, you know, these days, I don't know, if I were to say what's the scariest plot I saw last year, it was the energy usage in the United States, which doubled last year. And almost all that usage was related to AI and cryptocurrency. So literally, I don't think we would be talking about nuclear power the way that we're talking about this year, if not for AI and cryptocurrency, which is kind of a crazy thing to think about. At some point, I was telling my students who, you know, are worried about plastic bottled water, which is also a good thing to worry about, but they should also then definitely as an eco-friendly, earth-friendly way, they should also abstain from writing their essays with ChatGPT, because it's also something that has this capacity. And then you can think about, it's kind of interesting that transfer learning in a mechanistic model, it depends on evolutionary conservation, right? Not on the way that we typically think of transfer learning. And anyway, so there are these really interesting things. And then like what you were saying, James, is that we also have these big advantages, right? That there is a lot of data people can take advantage of right now. Maybe we don't understand how it all holds together, but we could still make predictions. You do have this ability to kind of like you were saying, in some ways you can gain insight a little bit faster. My favorite would be to include these two things together. So I would love to see, maybe some of you have seen is it Ross King who does the virtual scientists? Have you seen that? Where he has like an automated lab that he drives with some relatively small scale models, but he's trying to parameterize and predict at the same time. I would love to see, when I saw his thing talking about robot scientists, I immediately had another kind of money title come to mind, which is robot scientists studying virtual cells. Like if you had this model, for example, as a digital twin that you're just studying, you're studying it, right? Like you're just trying to figure out how it works. And could you use machine learning and possibly some of these really cool kind of experiment driving type of learning approaches to interrogate our system? Like one of the skeletons in the closet of everything that I've told you here is I can run simulations much faster than most people can run experiments. But when it comes to understanding what in the world I've just done, I'm as lost as anybody, right? So it takes us months to understand the simulations that we've run, but I think that time could be dramatically shortened because the mechanism is in there. Like I have the full mechanism included, and I'll even go as far to say that if you can't understand it in our model, you can't understand it anywhere. This is the easiest it should possibly be. And so again, positions wanted, right? People who can actually go in and start to learn from a virtual cell in an automated, learning driven way would be transformative.
Can I just build on that for a second? It's everything is in the model in a mechanistic way, but I feel like the challenge is now how do you consume all that information, because you suddenly have access, like real-time immediate access to all the values of all the pathways of all the different molecular processes and how do you ingest it in the human brain?
Yeah.
Right. Because when you're reducing it down, when you're doing experimental, wet lab experiments, you're reducing it down. So you're typically looking at, smaller slice at a time.
Yeah. Bingo. That is it. You defined it. It's I think that's the hardest part. So you could imagine so let's imagine now we're doing the like no holds barred nuclear option, right? I asked someone the other day if I gave them the code and all the papers that we used to build the model and whatever else they wanted, could they build a language model that could be interpretive for me the same way some people try right now to interpret their own medical records, which by the way, I do not recommend, but people are doing it. Could we find a way, because we have all that data to train on, it's kind of interesting because it's not only all the papers and all the documentation, but it's even the source code itself would all be available. I don't know if that would actually do anything or not. Some people say yes, some people say no.
Yeah, yeah.
I mean, one other direction to go, though, with the full models of the kind that you have, is asking the question, can you actually infer mechanism from measurable data? In the case of the E. coli, you have some time series in an experiment, but you don't necessarily know what the underlying mechanisms are. In your case, you've got a mechanistic model. you could look at the time series and then you could ask the question, can you, with appropriate AI, infer what the underlying equations governing the system were? Or is that information, is there too much ambiguity? Is there too much loss or too much noise in the system for you to be able to back out the time series? Now, of course, I suppose a lot of people associate that with Karniadakis's approaches, but this question of can you infer missing information mechanistically by looking at the residual between the prediction and the experiment is, I think, pretty important. And that can only really be answered definitively in the case of synthetic data, where you actually have an underlying system where you know what the right answer is before you start.
Yeah. I love this. Iran will remember that I had this open challenge in the lab, which no one has ever surmounted, but I think it's a big deal. So the idea is that not only does correlation not imply causality, but in fact, the opposite, which we almost never hear, is also true. Causality does not imply correlation, right? And so the challenge that I give people is, let's take the most fundamental causality in biology, DNA, RNA, protein, right? The most fundamental, and just make a Gillespie algorithm simulation of one gene, okay, that probably would not require more than 20 lines of code, right? You could easily write it in 1 hour. But what I would want is for someone to be able to take the resulting data and somehow automatically tell me that they have observed a protein being produced by an RNA produced by data, right? Those trajectories look they're so dynamic and they're so noisy that I really don't think anybody right now could just do it, right? So but it's so fundamental. It's like the unit operation of what we have to do if we're ever gonna solve these things. So anyway, if anyone wants a challenge, I would say that's a big one. And I'm happy to code you can even just look in my textbook, or I will give you a Gillespie algorithm, 30 line simulation. If you can derive causality from that, I would be amazed and astonished. Iran.
Am I still a candidate for this challenge? Can I?
Yes.
Okay.
Yes.
And are we allowed to manipulate the system, or are we just like running and only observing? Can we?
No, I'll allow perturbations.
Allow perturbations? Okay.
Yeah. What I think, I have to think it's absolutely impossible without the dynamics. But then you can't just like do a Fourier transform. You know, I think it's perplexing. I could even just imagine, I would totally be game to just sit around. This is the first time I've ever announced this challenge outside of the lab, by the way. Iran's heard it before, but you know, it would be fascinating. I would definitely, this is the kind of thing I could sit and talk about for hours, because it seems so simple, right? The Rubik's Cube seemed much harder when I was a kid. No one ever thought anyone would solve the Rubik's Cube, right? This sounds simpler, but it's much harder.
If you're willing to send us the description of the challenge with this code, we'd be happy to distribute it to the whole community. And
okay,
you can make it officially public, Marcus.
Yeah, that's fun. Okay, I will take you up on that. Let me summer. I have to be sitting on a lawn chair in the sunshine, and then I'll start thinking about it.
Sounds good.
I'm going to make one comment for Iran's point, which is, I think one of the things that we need to do, and you maybe have the resources and the talent to do it, or you and Iran, which is to have better mathematical descriptions of what perturbations are practical in experiment. In other words, we're always focusing, if you're a quantum physicist, of course, measurement is important. And so you have to be able to model measurement as well as the system being measured. But in biology, we tend to focus on modeling the bacterium, but not so much on describing what are the possible measurements we can make of it, or what are the possible perturbations.
Yeah.
And so to Iran's question, can you make a perturbation? Well, presumably if you could perturb everything, including the variables that are somehow inside of the system, you could probably determine the elasticities and things pretty well. But if you restrict to the experiments that are possible in the lab, then it's a much harder problem to interrogate the system. And I think we really lack mathematical and computational ways of describing possible experiments and formalism for describing those experiments at higher levels.
I love this question. So, this comment really. So I've actually talked to Agilent about this, believe it or not. So modeling the experiments is super important if you want to do like an apples to apples comparison of what you're doing. And a lot of these companies have incredible in-house, data on basically how, so could you use this? Could I perform an experiment with my model using kind of Iran's whole colony or population stuff where I'm simulating a flask? I'm removing a heterogeneous sample from the flask. I am extracting proteins, separating them on a gel and pulling them out and then illuminating that gel to see if I have a Western blot, like a simulated Western blot that really is, it kind of sounds crazy, but you're exactly right. Like we're simulating direct to bacterium and we're missing, for example, the variance that just comes from experimental noise, right? The variance that comes from different hands trying to do the work, right? So there's all this stuff. And I feel like these companies should have a vested interest. I even suggested at one point to Deep Origin, this company, that if you did something that was E. coli focused, you could do this really incredible thing where it exactly goes back to your thing. You make a map, because unlike a lot of large language models, et cetera, you could make a map. It's a finite number of experiments you can possibly do, even though it's a large number. It's a finite number. So you could make a map of every single experiment that is possible to do. And then, and what it does and what it finds out. And you could imagine making a really interesting network that I called your what should I do next tool.
Exactly, yes.
So I've done this, and this with hepatocytes or whatever. What should I do next? Well, have you tried a toxicology study? Oh, by the way, here's a link to the people who do it or any CROs. So it could be something that generated revenue. And then it's like, okay, once you've done this, upload your data. We will immediately find all the spots, do all the processing. If there's a model of the process beneath it, we can tell you how surprising it is. And then you could imagine, as it became global and as this thing was just aware of what everybody's doing, it could even say, 100 people are working on this exact same thing. Like maybe you'd like to try a new path, right? Maybe you'd like to try something different. I can make suggestions in that way too, right? I love this idea of kind of a discovery facilitator that kind of automatically takes these things into account. And it's years in the future, but it's really fun to think about. And I think it depends in some ways, if the model aspect was to realize its potential, it depends on exactly what you said, which is being able to simulate not only the subject of the experiment, but the mechanism of the experimental procedures.
So that's one end of it, which I think is critical. And I really appreciate the way you enunciated that clearly. The other one is maybe simpler, but at some level. But if we're going to do a sensitivity analysis mathematically, suppose we're doing an ODE, classic reaction kinetic model, then we can change parameters one at a time. We can change variables one at a time. If I use a drug to modify a biological system, it's going to change a lot of things at the same time.
Yeah.
And so instead of taking derivatives in our mathematical model in some known direction, we're on some manifold, which is the manifold of how that drug works in that system. And so we don't have total ability to manipulate independently parameters or variables in the system.
Yeah.
Now, so if we're doing molecular perturbations, we have an additional complication, which is that we don't have the ability to have clean control parameters. That absolutely the experimental conditions and the way the experiments are done on top of that makes it an even harder problem. But even at the lowest level, you may know molecular affinities from docking calculations, but fundamentally, when you put a drug into a system, it interacts with everything in the system. Ideally strongly with some things, then weakly with the things you don't care about. But it does interact with everything. And we don't really have a good understanding of how those perturbations across parameters work, I think. Now, the modeling that you're doing can answer those kind of questions. But I think for the most part, the kinds of models that most of us do don't try to get to that level of detail.
Yeah. I agree. And even in things we think we know classically, you know, just the way that a lot of data is reported, we don't get a lot of side effects, you know? So you're really kind of highlighting a huge gaping hole in a lot of the data that's out there. We tried this funny thing some years ago looking at phages, incorporating into E. coli, because I'd really love to put phages in there. I just love phages. And so we looked at Lambda, and of course, it's got a classical integration site, but we just looked to see with CRISPR, it was fairly easy to look and see if there were any more, and there were 300 more. And many of them kind of have oncogene-like effects in the sense that they actively disrupt something and actually change the phenotype of E. coli, right? And anyway, it was just such an interesting thing to realize, you know, these things that are going on that we just don't see. And I think maybe to your point, you know, maybe having a holistic model is really the only way to try to get at it, because otherwise, how are you going to wrap your head around that? It's, you know, we're kind of built to focus, which is a gift. I think it's good that we're built to focus, but it's as we try to get at the complexity that's kind of hiding behind all of our kind of simple stories that we tell about how cells work, you know, we probably need that additional thing. Yeah.
Okay. Well, I want to thank you for staying on and giving us a wonderful discussion. Thank you for the audience for staying on too and asking so many questions. And I hope we'll have you back soon, maybe talk about some of the many other things that you've worked on over the years and some of your new projects as well. So I want to thank everybody. We'll see people at our next meeting, and those of you who are on the steering committee, we'll see you tomorrow. Thanks again. Bye.
It was a huge pleasure. Thanks, everybody. Great to see you.
Thank you so much.
Take care.